Good morning, everyone. So, welcome and thank you for participating in this webinar. I'm Keat, Field Application Specialist in Apical Scientific. So, yes, today's webinar topic is Fundamental of Genomic Extraction. So, when I go through the slide, I will share some basic information and tips on the handling with you. And then if you have any questions, do type in the chat box. I will try my best to answer you after the slide sharing. So, but if anything that is specific to application, I might not able to answer you, or I might need some more information to search some information, then I will answer you through email later, or I may WhatsApp you. Okay, so and throughout this section, I will ask some question like what mentioned by Jenny. So anyone who give the answer the fastest and the correct, and we will give you a mystery gift. For every question, we will pick 3 winners. So do write your registration name and follow with the answer at the chat box. Okay, so not to delay, let us continue to the content for today. Okay. Okay, so in this webinar, first I will go through some introduction on gDNA and also DNA barcoding. Okay, then I will go into the detail on the genomic DNA extraction. And today I will specifically discuss on the detail on genomic DNA extraction from bacteria and fungus, which is the most common study sample in DNA barcoding application. Then within this section, I will share some general information with you, like why you need to use the chemical in the DNA extraction, and then there will be some tips on the handling also. Okay, then I will talk a little bit on the gDNA quantification and what is the result that you should expect in order for you to able to get the species that you are studying on when you perform result analysis. And in the conclusion, I will wrap up what is the tips and what I have shared with you and what you learn from this webinar. Okay, so not to delay, let's start with the introduction. Okay, so every life in on Earth is amazing, right? So they are unique creatures that will reflect from unique genetic information that inherit from generations. Okay, so all this genetic information are derived from genome, which encode by genomic DNA. So in short, we call it gDNA. Genomic DNA is the chromosomal DNA of an organism. It is an organic chemical molecule which construct by the deoxyribonucleic acid DNAs. There are 4 of them. So the A, adenine, T, thymine, C, cytosine, and G, guanine. Okay, so gDNA contains genes which will transcribe to produce DNAs, which are necessary for the function of the organism. Genome size very depends on the organism, so it can be ranging from a few kilobases in virus to a few megabases in bacteria, then to 10 to 100 of gigabase in higher eukaryotes cell. Okay, so human, we have estimated genome size of 3.2 billion base pair, which is 3.2 gigabase. So while plant, they have even more, that can be up to hundreds millions base pair of the genome. Okay, so where do you find genomic DNA? In eukaryote cell, usually we will get the genomic DNA in the nucleus of the cell, and it is usually a double-stranded DNA. While in plant cell, the genomic DNA is located in the nucleus also, but there are also some extra chromosomal DNA, which you can find in the chloroplast and mitochondrion. Okay, um, these two component, they contain part of the genetic material which are actually different from the one that is in the nucleus. Okay, okay. So in prokaryotes, the genomic DNA is composed of either single or double-stranded DNA, which is in the form of circular, and it is located in the nucleus of the bacteria. Okay. Some bacteria also carry a smaller extracellular extra chromosomal DNA called plasmid. So it's a uh, circular double-stranded DNA. And plasmid carry gene that may not necessary and not uh, essential for normal growth, but mostly it will help in the antibiotic resistance on in the bacteria. Okay, so plasmid, they can actually spread in the bacterial colony through the plasmid exchange. Okay, and then in virus, the genome can be either DNA or RNA. So we have DNA viruses and RNA viruses, and it can also be either single-stranded or double-stranded in the form of linear or circular format. So the it's kind of high variation can be found in the virus genome. Okay. Okay, so basically, genomic DNA play an important role in most of the biological research or molecular biological study. So you can say that it is the starting material for a study. And I will go through I will not go through every of the application here, but just an example in biotech. Okay, for example, genomic DNA is important, especially in molecular biotech application like cloning, genome editing, study of the whole genome, and study of uh, gene expression. Okay. Okay, also the genomic is important in drug discovery to check the drug response and therapy. In medical and uh, health application, to genomic is important for diagnostic and therapeutic decision making. Okay, so it's quite important, and also in vaccine development and gene therapy. Okay. While in the agriculture, plant genomic DNA is important in develop of transgenic crop and also enhance the food security of the world. Okay, so in marine monitoring, genomic study is the easiest and reliable taxonomy identification method, and it helps in assessment of the environmental status of the marine water. Okay, so this is also one of the objective of DNA barcoding, which we are going to talk in detail in this webinar. Okay, so what is DNA barcoding? DNA barcoding is a method used to identify, discriminate, and classify an organism. So it uses a short gene sequence from the genomic DNA, which we call it as a conserved gene for specific species identification. Okay, so this conserved gene can be found in the specific species. For example, like in bacteria, the conserved gene is 16S RNA gene, and this gene is not found in fungus or eukaryote. Okay, so that is why we can use the conserved gene to identify different species. Okay, so conserved gene usually is short, easy to amplify, and they have reasonable variation between species. So DNA barcoding is the standard research that is facilitate biodiversity study like species identification and discovery. So it helps researcher to understand genetic and evolutionary relation by the assembly the molecular and also morphological and distributional data. So then it helps in the marine monitoring. Okay. Okay. So here is the conserved gene which used in the DNA barcoding. And now it come for the first question for today. What is the conserved gene that used in the DNA barcoding to detect bacterial species? What is the name of the gene? The one that I did mention in the previous slide, just a few minutes ago, maybe in 1 minute ago. So please type your answer in the chat box and also your registration name so that we can know who you are. Okay. And let me check. Good morning. Me? So fast. Ashana Amin, 16S. Kashmira, 16S. And Chung Anchi, 16S. Yes, it's 16S. Tivaya also correct, but faster a bit next time. Okay, so we have the first three winner. Shuping also correct. 16S, yeah. So it's a 16S gene. Okay. Okay. So let me continue. So the conserved gene used in bacteria is 16S RNA. Yes, you all are correct. And to detect fungus, it is usually a ITS gene. Algae is 18S RNA gene. Animal is COI. Plant, there are actually two. It's either RBCL and another one is MAC. So actually some plant they have only one of this gene. So if you running a plant DNA barcoding, do try both of them. It will be better so that it is your study also. Okay, so all these conserved gene they present in the genomic DNA and is easily amplified by using genomic DNA as the template. So that's why in DNA barcoding, gDNA extraction is important. It is the one that carry the information that you want, and is that's why you want to get the information from this gDNA, right? Okay, so here is the workflow for DNA barcoding. There are 5 steps involved. So first will be the collection of raw sample. Then is DNA isolation. And one important point to remember is for DNA barcoding, you will need to make sure the sample is pure isolate so that you can get a good sequencing results in the end of the study, which will allow you to analyze and then identify the species. Okay, so if there is mixed isolate, then the sequencing result will be noise. Okay, then maybe you will ask me, my sample is mixed isolate, then how? For mixed isolate, you can actually go for amplicon seq under NGS application also. So from there you can know all the nearly all the species inside your sample. Okay, So for DNA barcoding, it is suitable for the researcher who want to study on some specific or to detect some specific bacteria or fungus. Okay. So in the second step in DNA isolation, this is the step where you get your starting material, the genomic DNA for your study. So after determine the gDNA, then you will need to perform PCR amplification to amplify and fish out the conserved gene of the specific organism by using the specific barcoding primer. Usually we call it as a universal primer. Then sequence the PCR product through single sequencing and analyze the result. It will end with blast of the sequencing result in some specific database. Usually we use NCBI in order to know what is the species that is detected in your sample. Okay, so as for today's webinar, we will concentrate on the gDNA extraction step, and we'll talk more on bacteria and fungus gDNA extraction. Okay, so under this section, I will have go through some introduction on gDNA extraction, and also you can say it's gDNA isolation is also the same. Okay. Okay, so for gDNA extraction, basically there are two methods used. One is the solution-based method and another one is solid phase isolation method. So for solution-based method, usually phenylchloroform isoamyl alcohol method, it will be used in extraction from bacterial cell, animal tissue, or blood sample. Okay, while the CTAP method is usually used in plant gDNA extraction. These two are the most known method for solution-based gDNA extraction. While for solid phase isolation, there are two methods also. One is using the silica membrane method, which we usually call it as silica spin column method. And another one is the magnetic base method. These two methods, they actually are nearly the same. Okay, so the principle are the same. Just one is involved in binding the DNA to a membrane, which is inside a column, and then another one is binding to magnetic base. Okay. So let's go through a little bit on each of the method. So for the principle in the solution-based isolation, for example, the phenylchloroform isoamyl alcohol method. Okay, so it involves first the lysis of the cell, then there will be a phase separation to separate the DNA and RNA and protein in the cell into different phase. So this is performed by mix of the cell lysate with the phenylchloroform in the ratio of 1:1. So from here, you will find there are three phases of solution in your tube. The upper layer usually is a aqueous phase which carrying DNA. Then the middle phase is the thin layer of the interface which carrying protein, while the bottom layer usually is the organic phase which carrying RNA and some lipid from the cell. Okay, so after the phase separation, you will need to pipette out the aqueous layer which actually carrying DNA into another tube and then proceed to DNA precipitation by using ethanol or isopropanol. So after that, there will be washing of the DNA with ethanol and dissolve of the DNA so that you can get the clean genomic DNA from here. Okay, and one advantage for this method, remember there is the other two phases from the phase separation. So you can actually isolate RNA and protein from the same sample from the interphase and the organic phase. But there is a drawback for this method also, because when you want to pipette out the upper phase, okay, sometimes you may mistakenly take some of the interface or some of the organic phase into the new tube. So this is how it causes the contamination. Okay, else maybe you will say, I will leave a little bit here. So you can leave a little bit here, but if for the sample that they are quite low in the DNA amount, then it will causing the yield of the DNA extracted will be even lower. Okay, so that is the advantage and also the drawback of this method. So to avoid the possible of yield and purity problem, nowadays the solid phase isolation method is more favorable. So in solid phase isolation, first it will involve the lysis of the cell, then continue with the binding of the DNA to the solid phase, okay, which is the silica spin column or the magnetic beads. Then the principle is actually quite simple. The DNA will bind to the silica membrane or the magnetic beads in the presence of high concentration of chirotrophic salt. And the contaminants like protein and RNA, they will be washed away. So you can get the DNA later elute in the water or the low salt buffer. Okay, so one advantage of the silica membrane spin column is that silica is bound to a solid support, which is the column. So with this, it actually eliminates the problem of glass bead contamination in the extracted DNA. Okay, so I actually found some papers stating that spin column method is more favorable for example, in this review. Okay, in this review, it actually stated that the column-based nucleic acid extraction is one of the best technique uh, among the options available, playing a vital role in ion exchange methods. So it provides a robust stationary phase for a rapid and reliable buffer exchange. So it helps in nucleic acid extraction. Okay, so this method is fast and reproducible. um, One drawback is you need a small centrifuge. Okay, so I believe this is not a problem for molecular lab. So certainly, nearly all molecular lab will have centrifuge, right? Okay, so for DNA barcoding application, in order to perform a sensitive, specific, and reliable PCR based assay, then the availability of the pure DNA and as well as easy perform DNA extraction is very essential. Okay, so today we are going to talk in detail on spin column method. So now please allow me to introduce you the Primeway genomic DNA extraction kit from first base. It uses silica spin column method for the gDNA extraction, and more important, it is suitable for extraction of a wide range of sample type. So especially is very useful for DNA barcoding, because sometimes I believe for DNA barcoding lab, usually you will have sharing between some of your colleagues that some of them is used studying on maybe bacteria, some will be studying on fungus or um, um, fish. So this kit certainly will help you. Okay. Okay, so first let me share some highlight on this kit. Primeway Genomic DNA Extraction Kit is flexible and it can use for 10 sample types, which include human or animal tissue, cultured cell, mouse or rat tails, bacteria of gram-negative and gram-positive, yeast and fungus, also blood on FDA card or whole blood or from Buffy code. And also you can extract DNA from insect and buccal swab using this kit. Um, there are some specific protocol to help you determine maximum yield and purity, especially designed for each of the sample type. So for there will be 10 protocol which is specific for this 10 sample type. Okay, so the extracted gDNA is good in quality and is suitable for PCR, restriction enzyme analysis, certain blot, and also you can actually get a NGS grade of human gDNA by extract from Buffy code of the blood. So we have a specific design protocol for this NGS grade human gDNA extraction from the Buffy code. Okay, so maybe you can scan the QR code here to have a detailed view on the full protocol, because later upon the webinar session, so I will talk a little bit on the protocol. So while we go through the protocol, then I will share the tips and the basic information with you. Okay. Okay. But there is many sample type here, right? So today we will be discuss on bacteria and fungus. So if you are interested in other sample type like human or animal tissue or blood, so or some culture cell, you can type on the chat box and on your request. So maybe we can consider to have another section on the webinar of different sample type. Okay. Okay. So again, the workflow for gDNA extraction using silica spin column, and okay, basically you will find that for DNA binding, washing, and elution step, they are nearly the same in all of the sample type and also in most of the commercial kit, um, as I show here. Okay, in these three steps, ethanol will be used to facilitate the binding of the DNA. Then it will continue with the washing of the DNA for twice, but it's kind of depend on the kit also. And then elute the DNA with elution buffer. Okay, so these three method is the same in the um, in nearly all the sample type and commercial kit. Okay, so what is the difference? The difference is usually at the sample preparation and the lysis step. Okay, for different sample type, they have some different in the handling of sample preparation and lysis. Okay, why? Because lysis of a cell is very much depending on the structure of the membrane of cell. So like bacterial cell and animal cell, they have some different. Even positive and gram-negative bacteria also have different, right? So plant cell, they have even much more different. and much more difficult to lyse. Okay, so we need some specific methods to break the membrane of the cell. Okay, so today we will going to talk about bacteria and fungus. So we need to understand a little bit on the membrane structure, where it's the place where you want to break the cell, right? So in gram-negative bacteria, in the membrane cell, they are surrounded by a thin peptidoglycan together with an outer membrane cell, which contain of lipopolysaccharide. And meanwhile, in the gram-positive bacteria, they do not have the outer membrane cell, but they have a very thick layer of peptidoglycan, and this thick layer is hard to break, and so we actually call it as a cell wall for the gram-positive bacteria. Okay, so the peptidoglycan are actually a long anionic polymer. So it need to be break down by hydrolysis with enzyme called lysozyme. So that's why for lysis of the gram-positive bacteria, you may find some additional treatment step is needed with this lysozyme. Okay. As for fungus cell, the membrane cell is predominantly made up of sterols and some phospholipids. Okay, while the cell wall, they also contain of a protective layer of chitin and also glucan on the surface. Okay, and okay, surface there is a manoprotein. Okay, so all these layer, they actually um, make the cell of the fungus difficult to be break. And in the different fungus cell wall, they actually contain different glucan. So for example, in the cell wall of Aspergillus, they contain beta 13, beta 14, and also alpha 13 glucan. And while you find in Canada, they actually contain only the beta 13 and beta 16 glucan. So the glucan layer here, they can be quite different. Okay, so it make it um, even much more difficult for you to optimize the lysis process. Okay, so now we go into the process of the gDNA extraction. Okay, so first, do remember for DNA barcoding, we need a pure isolate. Okay, so how to get a pure isolate? You can use pour plate, spread plate, and strip plate techniques. Okay, but I'm not going to discuss on this technique in detail because the time is quite limited here. So next, after you determine a pure isolate for bacteria and fungus, we'll go into the gDNA extraction step. Before I talk in detail in the process of extraction through the protocol sharing, okay, here is the second question for today. What is the extraction method used in Primeway Genomic DNA extraction kit? I did mention for the past few slides quite often, so please type your answer in the chat box. Let me check in the chat box here. Silica spin column method. Yes, Muhammad no Adam is correct. Solid phase method. Yes, is especially the column method. Yes, you can say it's correct also. Clarissa N. And spin column, Muhammad Rizal. Silica spin column method. Yes, it's silica spin column method. Most of you is correct. Wow, you all pay attention in the webinar. Oh? Very good. I'm so happy. Yes, 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 you all are correct. So it's silica spin column method. And the okay, so okay, we continue. For bacteria, the sample preparation for extraction is quite simple. So you actually only need to prepare the cell pellet by centrifuge and remove the supernatant, which is usually the culture medium. Okay, and the cell will be ready for the next step, the lysis process. While in fungus, there is an additional step to wash the pellet with the EDTA in order to remove contaminant. Okay, why EDTA? Because the high affinity and strong collecting property of the EDTA, it actually allowed the complete and irreversible denaturation of the metal containing proteins. So it actually helped to improve the purity of the DNA extracted. Okay, so this is especially important for soil sample because soil samples sometimes they have heavy metal. Okay, and I can say maybe for sample that is determined the water sample from ocean maybe because now our ocean is contaminated with heavy metal also, right? So yeah, all this sample is important to pre-wash with EDTA and then centrifuge and remove the supernatant, then the cell is ready for lysis. Okay, okay. In the lysis of the cell, as mentioned just now, different cell they have different component which build up the membrane cell and the cell wall. So a good lysis buffer is very important in DNA extraction kit. In Primeway kit, if any of you did scan the QR code just now, so you can go to the protocol and you will find the kit content inside. So there is two lysis buffer if you look into the protocol, and actually some of the sample type can be lysed using one of the buffer, but there is also some that need both lysis buffer. Okay, so upon the lysis step, you will actually find that proteinase K is always required in all the commercial kit. Okay, it's not only in Primeway, but it's all of the commercial kit because this enzyme is very important in DNA extraction. It is a broad spectrum serine protease. So proteinase K, it helps to digest protein in the cell lysate so that the DNA extracted do not have the protein contamination. And with this function also, it actually means that proteinase K, it will digest nuclease. Okay, so nuclease is the one that will be degrade the DNA. If you're doing on RNA, is also degrade RNA. Okay, so in the other words, it means that this enzyme protect DNA from degradation upon the DNA extraction. Okay, so in primary kit, we have proteinase K and the buffer ready, and the step for the use of proteinase K is actually quite easy because we combine the treatment of proteinase K together in the lysis step already. So it makes the protocol much simpler. Okay, here just to share some additional information on proteinase K. The working condition of proteinase K in DNA extraction is usually at around 50 to 400 microgram per mil. So most of the commercial kit, they will use 20 to 25 microliter of the 10 milligram per mil proteinase K. So it actually gives a working concentration of about 200 microgram per mil. So this condition is quite optimized already. Yeah. And then for the storage, do remember the storage of proteinase K is very important because you want to preserve the function of the proteinase K, right? So proteinase K, it is stable at room temperature for 1 year before you mix with the buffer. But after resuspend in buffer, it is advisable for you to store it at -20 for and you need to use it within 6 months. It's highly advisable. Okay, so that you can make sure the function it is functioning well. Okay, so in the lysis step, you will find that there is some additional step needed for gram-positive bacteria and also fungi cell. So in the gram-negative bacteria, the lysis step is quite short, but in gram-positive and fungus, there is some additional step. So I will talk about this additional step with you. Okay, so in the lysis of gram-positive bacteria, lysozyme was used. So actually, lysozyme is a lytic enzyme which able to break down the thick cell wall of gram-positive bacteria, which composed of peptidoglycan, and it will disrupt the bacterial membrane. And as this bacterial type is difficult to lyse, we also need a pre-wash with the cell with the bacterial pre-lysis buffer. So this buffer, it actually contains the Triton X100, which is a commonly used detergent to lyse the cell. Okay, so this buffer, yeah, is not provided in the kit, but we do provide the formulation for you. So it's easy to prepare the buffer. Okay, so here if you do not have lysozyme, okay, some of you may say, I don't want, I don't have lysozyme with me, but now I'm already in the step, my bacterial gram-positive bacterial cell already ready. So can I proceed with extraction? The answer is yes. Actually, you can. Lysozyme it's actually can help you to get higher yield for your extraction, but if it is for DNA barcoding or if your application is not need so high of the gDNA yield, then you can actually skip this step. But somehow some different type of the gram-positive bacteria, they may have different thickness of the cell wall. So if you find that your gram-positive bacteria, after you skip this step, you cannot get I mean the yield that you determine is very very low and you cannot get the good amplification out of the gDNA extracted, then it is advisable for you to consider to perform this step. Okay. Okay, so in lysis of fungus cell, sorry, the cell wall will need to pre-wash with sorbitol buffer. So sorbitol is actually an osmotically active sugar alcohol, so it does not pass the cell membrane and likely to act by drawing the cytosol out of the cell. So therefore, it actually can remove polyphenol and polysaccharide from the cell wall and the cell membrane. Okay, so sometimes when you extract from fungus, you may find that the cell lysate showing some brown color. Um, so by pre-wash with the sorbitol buffer, it actually can help you to remove all this color. Although actually this color, they will not inhibit your PCR, especially for PCR, they will not inhibit it. So um, you you can choose to just proceed even there is some color in your sample. Okay, so also under this step, if you found that the first supernatant is cloudy, then you may repeat the sorbitol washing step. But usually one time of the washing is more than enough, especially for DNA barcoding sample. Okay, so this buffer, you can find that is not provided in the kit. In most of the kit also. Why? Because sorbitol is a sugar, so it actually need to be freshly prepared. So if you need sorbitol buffer, you can order from us, then we will freshly manufacture it for you upon your ordering. Okay. Okay. So in the lysis of fungus cell also, there are an additional step to use zymolylase in the lysis of the cell. So this enzyme is actually a lytic enzyme also. It its activity is to hydrolyze the glucose polymer at the beta-1,3 glucan linkage. So it actually breaks down the cell wall of the fungus cell. As what I mentioned just now, there is many different glucan in the fungus, and actually most of them, they carry this beta-1,3 glucan. So this enzyme will help you in determine a better yield for your fungus extraction. Okay. Okay. So next is under the protocol of Primeway Keat, you may also find that there is an additional step to treat the cell with RNAs. Not only under this kit, actually you can find it in most of the kit also, but usually the RNase treatment is an additional step lah. Yeah, so RNase as I believe most of you know, is an enzyme to degrade RNA, and this step it's needed if the application need a high purity DNA. But in DNA barcoding, I would say a little bit of the RNA, it will not affect your PCR. So you may choose not to perform this optional step for DNA barcoding application. But if you find that after extraction there is still many RNA, and then you want to treat with RNase treatment, is also can, right? Okay. Okay. Here in the lysis, you also will find that the incubation for the lysis process should be at 56 degrees Celsius. The temperature need to be at 56, but the period can be around 1 to 3 hours or overnight. So for DNA barcoding, because the gDNA will be used as a template for PCR, so you actually no need to have too high of the gDNA content, because if you have too high, it can sometimes causing some unspecific amplification also. So later on, if your gDNA is too high in the content, you still need to dilute it. Okay, so lysis of 1 hour should be sufficient for DNA barcoding application. But as mentioned just now, fungus membrane cell, they are quite varied. So you may need to take some time to optimize the period of incubation for different type of fungus. Okay, so this is actually applicable for on most of the commercial kit that you are using. So for fungus, you need to optimize, uh, make some optimization on the incubation time. Okay, and one more additional tips. If you are if you have done the RNA treatment but still found that there is RNAs in your sample, do double check the condition of your RNAs. There is a few cases that we find something like this. So do store at your RNA at the minus 20 degrees Celsius, and then actually the shelf life for RNAs is up to 6 months only. So make sure you keep it well. Okay, sorry, our line here, the line broken. Okay, so after the lysis of cell, okay, so can you all hear me? Okay, so after the lysis of the cell, it will continue to the DNA binding to the silica membrane in the spin column. Okay, in most of the commercial kit, you will find that they most of them is using ethanol. So as mentioned in previous slide, if you still remember, I believe all of you still may remember. You all very concentrate one. So the principle for spin column is actually the DNA will bind to the silica membrane in the presence of high concentration of the chirotrophic salt. So this chirotrophic salt, it can be phenol, it can be ethanol, it can be guanidine hydrochloride, urea, and also lithium perchloride. Okay, so ethanol is one of it. So that's why it will facilitate the binding of the DNA to the silica membrane in the spin column. You may ask me why, but I won't go into too detail on the mechanism. So it is a bit difficult to explain, but just a simple one. DNA is actually hydrophilic, right? So it will form a hydration shell in the water. So when it forms this hydration shell, then it will avoid the DNA from binding to the silica membrane. So this chirotrophic salt, it actually will weaken the ionic bond within the DNA and the water molecule. So when it breaks down, it means that the DNA will be freely in the solution. So it will bind to the enhance the binding of the DNA to the silica membrane. Okay, so here I would like to share with you some tips. For ethanol, there is denatured ethanol and non-denatured ethanol. So denatured ethanol actually they contain some additive such as methanol and isopropanol, which some of it may affect your downstream application. So for DNA extraction or nucleic acid extraction, you should use a non-denatured ethanol. Okay, so remember to ask for denatured ethanol when you want to do the ordering. And some of you may ask me, there is many grade of ethanol, so which one is better? Do look for a biotech grade or molecular grade. So of the ethanol, then it's good to be used. Okay. And additionally, okay, ethanol is actually a danger goods. It is category as a danger goods, so a DG item. So you'll find that most of the commercial kit they won't include it. Okay, so you need to order it, and then only they will prepare for you. So because the commercial kit, they will have difficulty in shipment of the kit and also to get the license if they include the ethanol inside. Yeah, so most of the kit won't have the ethanol. Okay, so yeah, for washing steps, uh, usually washing step they generally contain alcohol to in order to remove protein, salt, and also other contaminants from the sample. Some kit only use one washing steps uh, with ethanol, because ethanol it allows the salt to dissolve and minimizing the DNA solubility. In Primeway, we actually use two washing buffer, as one of the washing buffer actually carrying isopropanol. Okay, isopropanol, the wash buffer T1, okay, is actually carrying isopropanol. So isopropanol, it can work better than ethanol to precipitate protein. So it helps to remove protein from the DNA. And with the second wash buffer, which carry ethanol, it helps to eliminate salt. So with these two methods, these two washing steps, then it help you to clean away all the protein and salt, and make sure your DNA extracted is high in purity. Okay. Um. Okay. In the drying step, is very important. um, do make sure in the drying step, the wash buffer is completely removed, because wash buffer they carry ethanol which may inhibit the PCR. Okay, so um, if you perform the washing steps after centrifuge for 1 minute, you still find some remaining ethanol on the cap of the column, or at the below of the column, then you may need to centrifuge it again. So centrifuge for another 2 to 3 minutes will be enough. Okay, so not to centrifuge for too long also. And in elution step, uh, yeah, you need to uh, elute, put the elution buffer at the center of the column, so that it will make sure the elution buffer has covered the whole column. Then it will increase the recovery rate. Okay, but do not poke the column and do not uh, break it. Okay, so yeah, as for the volume of the elution buffer, okay, I will say always follow the volume recommended by the kit. I understand. Some of you may want to increase the concentration. You can reduce a little bit on the elution buffer. You can reduce a little bit, but try not to reduce too much, because reduce from our tests upon our manufacturing of the kit, we actually know that if you elute with too low of the elution buffer, then it actually will sacrifice the recovery rate. um, the eluted, the concentration may be increased, but the the recovery rate will be lower. Okay. Okay, so now you have complete your gDNA extraction. What you should do? Okay, usually there are two methods. First is to check with nanodrop or spectrophotometer. Pure DNA should give you a purity at absorbance ratio of 260-280 around 1.8 to 2, while the ratio for 260-230 is around 2 to 2.0. Okay, then you should run a gel to quantify your gDNA. There will be some smear found, but uh, as long as you can observe a high molecular weight band, this band, then it is good. Okay, so it means your gDNA extracted is good. Okay, so now it comes the third question for today. The gDNA extracted here is using which kit? Okay, so please state the full name of the kit and please type your answer in the chat box. Let me see. Let me check. Chat box. Oops. Any answer? Primary genomic DNA extraction kit. Yes. Primary kit. Primary spin column. Yes. Primary. But primary is spin column. Yes. But the kit, the full name of the kit is actually uh, Kuchi Chang is correct. Okay. So yeah, I not to delay. I will go into the slide again. So it's primary genomic DNA extraction kit. Okay. So after DNA extraction, you will have your gDNA. Okay. So as a template for your PCR, so for barcoding, DNA barcoding, then you need to perform the PCR with the specific universal barcoding primer. For example, for 16S, the well-known universal primer is the 27F and 1492R. And also for ITS is ITS 1 and 4. Okay, it's just a simple endpoint PCR. Okay, so after PCR, you need to quantify your gel, quantify your PCR product with gel, and then make sure that it's single band, and then before you send for sequencing. Okay, so sequencing is a Sanger sequencing method, a first generation sequencing method, but now everything is automated. Okay, so we are using strand terminator chemistry for the Sanger sequencing. After sequencing, you need to perform alignment and blast. Okay. Okay, a little bit on the result analysis. For DNA barcoding, you should sequence your sample with both forward and reverse primer, because some of the customer, they will ask, do I need to sequence both direction or only one direction? Yes, you actually need to sequence for both direction, because after that you can align the sequencing result and then get a full length or nearly full length sequence of your sample, then you can get a better blast result. Okay, so a little bit on sequencing result. You need to make sure the peak of the sequencing result is sharp and evenly placed with little or no background noise. So if ITS, the product size is around 700, so you should actually find the sequencing result end with a A here. So it is a stop point generated by the polymerase in the sequencing reaction. Okay, but in 16S gene, then you will not see this, because the full length, the PCR product size is 1.5 kb. But sequencing until now, single sequencing, they can actually read up around 1 kb, or if you're lucky, it may be 1.1 or 1.2 kb like that. So you won't see a stop point there. Okay, so the raw data of the sequencing result, it should have a nice baseline. So if you find the baseline is flying, then it means there is salt contamination. Okay, and in every of the sequencing result, you will find some loss of the signal in the front part. For your information, is actually just due to the loss of some extract extension fragment upon the post-sequencing purification, because purification, it will certainly causing some loss, especially a short fragment DNA. Okay, so it is normal. Okay, so once you determine the sequencing result, what you need to do, need to perform sequence trimming and then align both forward and reverse result. Usually use the pairwise alignment and then get a full length of or nearly full length sequence and blast in database. So usually it's an NCBI database. Okay, so from here, you know your sample is similar to which species. Okay, so yeah, because of the time, I won't go into too detail. If any of you request result analysis part for another webinar, you can also type in the chat box. So let us know what you need for the next topic. Then maybe we will prepare it for you. Okay. Okay. So conclusion. In this webinar, I've concentrated on the detail on genomic extraction from bacteria and fungus for DNA barcoding application. So in short, for DNA barcoding, you need to make sure the sample is pure isolate. So you can determine this through pour plate, spread plate, and strip plate techniques. Okay, you can Google. And the membrane and the cell wall of different sample type, they are actually have different. So a lysis step in the extraction is very important. Do make sure you use the proteinase K in the lysis step, and also make sure you store this enzyme. Treat it properly, okay? So store it properly, handle it properly, so it can do a good work for you. For gram-positive bacteria, lysozyme and pre-lysis buffer is needed. However, if you do not have lysozyme, you can skip this step when you're using our Primeway kit, because we actually tested it. We have the data. So the DNA amount if you skip this step also, it should be sufficient for DNA barcoding application. Okay. The lysis period normally is 1 to 3 hours. It should be sufficient for DNA barcoding. But if you need higher yield or with a complete lysis, then you may go for overnight lysis. For fungus, you may need to optimize the lysis period because the cell wall of fungus is vary between the fungus cell, different species. Okay, ethanol is important in gDNA extraction for binding DNA and also for washing. So make sure you use non-denatured ethanol, and a biotech grade is good. And do make sure the column is dry before you add the elution buffer, and make sure you put the elution buffer at the center of the column to get a full recovery. Okay, so under today's webinar, you should also know how to quantify and what is the method to use to quantify a gDNA. There's two methods: spectral photometer and agarose gel electrophoresis. Then I believe you have learned a little bit on what to expect in your sequencing results. Okay, so but I'm unable to go into detail on the trimming alignment that. Okay, so maybe future we can have a chance on this also. Okay, so from today's webinar, we know that good extraction kit is always will ease your molecular work. Okay, so under Primeway, a little bit on the advertisement. Okay, so actually we have a series of kit, not only the primary genomic DNA extraction kit. So we have genomic DNA extraction kit for 10 sample type. We have also if you are working on plasmid extraction, we have plasmid extraction kit. If you are working on PCR, then you need to purify your PCR. We have gel extraction and PCR purification kit. Also, if you're working on plant or virus, then we have the kit specific for plant and also for viral DNA RNA extraction kit. Okay. So yeah, grab it now because we actually have the promotion for 35% off for all primary kit. All primary kit. So until end of this month only. So grab it fast. Contact us fast. So okay. One more bonus question. Uh, what is the kit's name that can extract 10 different sample type? What is the kit name from primary? Okay, you all can type the answer in the chat box. I will just go into the next slide. Yeah, here is the answer. Primary genomic DNA extraction kit. Okay, so I end my presentation here with a video. We'll go into the Q&A section later. Okay, after the video. Let me play the video. Wait, sorry. Hey, why I cannot play the video? Moment, I have problem to play the video. Hi, sorry. Okay. It's my hand.